make sure that you keep making people coming.
All right, welcome everybody to our VacCAP webinar. We are really excited to have Dr. Massimo Iorizzo today as our speaker. Just a reminder to everybody, please keep your microphones off, muted. If you can keep your camera also off, that would be great. We are recording this and we're going to post it on our YouTube channel for our project. And so we'll put the link in the chat. If you have any questions, you can type them in the chat. At the end of the presentation, we're gonna go through the questions, but feel free to just add all the questions that you have as Dr. Iorizzo is presenting. So with that, let me introduce Dr. Iorizzo, who is an associate professor at North Carolina State University in the Department of Horticultural Sciences, and also is part of the Plant for Human Health Institute. And he is the project director for the VacCAP project that is funded by USDA NIFA. His research focuses on structural genomics and genetics for quality-related traits in fruits and vegetable crops. In 2016, Massimo initiated efforts to develop a community-based project for vaccinum crops, which led to the development of the VacCAP project. The title of the webinar, and I'm going to say, Massimo, you really pick a mouthful here with this title, because it's very difficult to get it all in one phrase, but it's Comparative Genome Analysis in Blueberry Revealed Autopolyploid Recombination Behavior and Heterozygous Reciprocal Translocation, which represents a collaborative study that was recently published and contribute to the advanced multiple research efforts in blueberry genetics and genomics. And with that, I'm going to let Massimo take over. Again, please make sure that your microphones are muted so we don't interrupt Massimo during his presentation.
Okay. Maya, thank you so much. Let me share my screen. Here. Okay, can you see the slide?
Yes.
Okay, and then I mean that you can hear me. Okay, thank you, Amaya, for the introduction. So, as Amaya said, we'll talk about blueberry genetics and genomics today. The talk of today, it's part of the VacCAP project, and the project focuses on heavily on quality, like genetics of quality trait, and develop around 5 objectives. But the research that we will present, that I will present today, it's part of the objective 1, which is the focus of this objective is to establish genomic and genotyping resources through comparative genomic analysis, so developing new genomes for blueberry and cranberry, and use this information to develop a genotyping platform. So the results of the webinar today are largely published in this article that was recently released in New Phytologists. So the outline of the talk is development and characterization of a new blueberry reference genome assembly, comparative analysis between tetraploid and diploid genomes, and elucidating the recombination behavior of blueberry. So the first part, it's about a new reference genome for blueberry, which it's the W85 genome. It's representing a wild species, Vaccinum caesariensis, which is also named Vaccinum corimbosum, or New Jersey high bush blueberry. It's diploid, and multiple studies have reported that this species is morphological and not distinguishable from Vaccinum corimbosum. Based on morphological characteristics, crossability and geographic distribution has been proposed as a potential progenitor of Chromosome. And based on some molecular marker work recently published has been highlighted that it's the closest relative species to Vaccinium corymbosum. So there is high similarity with the Vaccinium corymbosum, and that's why it was selected as a genotype for sequencing. So the efforts, so the genome was sequenced using long reads based platform, PacBio sequencing, integrated with Chicago, Iorizzo, and High-C proximity, Illumina, RNA-seq, and PacBio IsoSeq for gene prediction. So then we did multiple analysis to evaluate the quality of the genome assembly, and we will see those analysis. And then we used a newly developed linkage map that was recently published and include over 17,000 SNPs markers for anchoring and verify the quality of the genome. So the genome was assembled at haplotype level, which means that for each of the chromosomes, we were able to assemble both haplotypes. So then we have 12 chromosomes, 24 haplotype. These are some basic statistics of the genome assembly. It covers about 100% of the estimated genome size. Large part of the genome, over 97%, was anchored to chromosome level. Then the contigent 50, it's about 0.4 megabase. 45% of the genome is represented by repetitive sequence, and then we predicted about 34,000 genes in both phase 1 and phase 2. Those are the two haplotypes, and most of these genes were anchored at chromosome level. So just as a reminder, we already have a nice reference genome for blueberry, the Draper genome. Compared to that genome, this genome has about 10-fold higher contigent 50, so higher contiguity, and then the longest haplotype had 106 megabase into the sequence, about 22% of the total longest haplotype of the Draper genome, which make a great complementation to that available genome. So as part of the process of assembling the genome, we made a lot of efforts to verify the quality of the genome assembly, the contiguity of the contig and the scaffolds at the chromosome level and contig level. So here it's a representation of comparison between the linkage map that we used for anchoring and verify the quality of the genome and the chromosome 2 assembly. We can see that there was a rearrangement or a misassembly. Here it's the IC map, heat map that showed that there is a discordance in this region of the assembly. And then we went closer into the assembled sequence, and we could find that there are regions at that junction that are not covered by link of parent data and neither PacBio reads. This is likely a chimeric assembly that we broke and then we reconnected correctly in the assembly. So then a number of those chimeric regions were corrected, and finally we got our final assembly that it's highly collinear to the linkage map. Here it's the example of the correction of the chromosome 2 that we made that now became completely collinear, and here it's the profile of the IC map, which it's the desired or expected profile with a large number, with a straight line to follow the interaction between the link that were made using the IC data. So then we did some QC analysis for verifying the gene content and how much of the repetitive sequence were covered, correctly assembled. So we did two analysis, BUSCO and gene space analysis. Using this approach, we saw that a large fraction of the gene represented in other species are conserved gene are represented in the genome. We mapped the transcriptome data back to the genome, and then over 99% map back to the genome. Then we did an analysis that is called the LAI, which look at the presence of intact heterorasposome element, and then these index are relatively high for this genome. Then we annotated over 97% of the gene. This demonstrated that the genome cover most of the gene space and repetitive sequence. Next, we started to look at analyzing or mine centromeric region in the genome. So centromere in the genome are critical because they are important for the genome to have a normal segregation and duplication of the chromosome, and play an important role into the divergence and speciation of the genome. Now, how we looked at this, we focused specifically on some structural feature of that the centromere usually have, and these include they are enriched in a tandem repetitive sequence. Usually, they tend to have cover region with low recombination frequency, and they are rich in gypsy transposable elements. We started using this approach to look for this feature in the genome for identifying possible centromeric repeat in the genome. So this is a scheme of the assembly of the 12 chromosome, and here we can see we identified two satellite repeat, one called VacCAP1 that is 147 nucleotide, and VacCAP 169 that is 480 nucleotide. This is what looks like VacCAP1, these 147 nucleotide that are repeated many times next to each other. So then we mapped those satellite repeat in the genome, and what we noticed is that VacCAP1 actually it's located in the region that have very low recombination frequency in linkage maps, and then have a high abundance of LTR gypsy, which are the characteristics of a possible centromeric repeat. It's noticeable that this repeat was present in almost all the chromosomes, and the VacCAP 169 was specific of chromosome 6. So then next we did some further analysis using fish analysis. So we designed a probe specific for this 147 base pair and this 480 nucleotide repeat. Then what we noted is that as expected, we obtained 20 chromosomes that had the red signals, 2 chromosomes that had the signal specific for VacCAP 169, and then there were 2 chromosomes that had no signal, which are these 2 chromosomes here. This chromosome here that has no trace of this repetitive sequence. Most importantly, when we looked at the metaphase 1, we could see that the satellite VacCAP1 actually it's coincide with the pole of the chromosome separation, so which indicate that the satellite is associated with the fuse of the chromosome where they are pulled in during the meiosis. So it's an indication that these are associated with the centromere. Then we did an analysis of the cytogenetic work on the tetraploid samples, and then we verified that also in the tetraploid we found the 20 signals with the VacCAP1, the 2 signals with the presence of the VacCAP 169 that is specific of chromosome 6, and then the 2 chromosomes that have no specific of this satellite repeat. Then we looked at the conservation of this satellite repeat in other species, in particular in Vaccinium myrtillus and Vaccinium darwii, which genomes are available. We could see that the VacCAP1 is highly conserved in Corymbosum, tetraploid Corymbosum, and diploid darwii and caesariensis. So this is the phylogenetic analysis, which indicate that these 3 species have a very conserved motif of this satellite, which is distinguished from Myrtillus. VacCAP also has the same distribution of this satellite 1 and satellite 169 as W85, expected that its chromosome 6 has a high abundance of the VacSat 169. And then when we did the fish work, that matched this expected profile. So as part of the assembly, like I said, we did the genome is assembled at haplotype level, and then we were able to characterize the two haplotype. So we found that about 1.7% of intragenomic variation, 47% of the gene harbor a potential deleterious mutation, and more than 2% of the genes were present absent variant genes. And then path genes were enriched in resistant genes, which has been observed also in some other species. So now, having this high quality genome, ensuring that we have a good established framework on the genome, we then started to use the genome and other resources like linkage map to start to make some comparative analysis with tetraploid genomes. So for this analysis, basically what we were trying to understand is the structure of the diploid and tetraploid genomes conserved? And another question was that we want to answer if there is some structural divergence, like was previously noted in previous study, where they found a possible translocation between chromosome 6 and chromosome 7 and chromosome 10 in the Draper genome. So then we verify the presence of this translocation, and it does affect the recombination behavior of the tetraploid plants. And how conserved is this translocation in other species, in other tetraploid plants. So to do this analysis, we developed 3 new linkage map representing 6 cultivars, which all harbor from 10 to 60% of wild genome. So the 3 linkage map are named R by A, DS by J, D by B, and one of them, D by B, should have technically should have the translocation because one of the parent it's Draper. Then we also used the Draper genome that is available. Then we used all these resources to compare with the W85 genome. So the comparative analysis overall indicate that the W85 genome and the tetraploid genomes that are part of the linkage map are highly collinear. The comparison with the Draper genome also highlight high level of collinearity, although in some cases what we found is that there were some possible misassembly in the Draper genome. Here we can see this part of chromosome 9 that has a possible rearrangement in comparison with the Draper genome. But when we compare this region with the linkage maps, with 2 linkage maps, we can see that that region is completely collinear. This is likely the results of a chimeric region in the Draper genome assembly and not a real biological rearrangement. And the same, it's this example here where it's collinear with the other linkage maps that are tetraploid and not with the Draper genome. So then what about the translocation that was previously identified but requires some further analysis to verify the presence of this translocation? Like I said, there was a translocation identified between chromosome 6 and 10 in the Draper genome, and then when we compared with the W85 genome, here we can see W85 chromosome 10 and W85 chromosome 6. It's completely collinear with the 2 copy of chromosome 6 here and 2 copy of chromosome 10, and then it's completely, but as a rearrangement between chromosome 6 and chromosome 10. So there is a translocation, a piece that is translocated from chromosome 10 to merged with chromosome 6 here, and we call this chromosome 610. And then on the other way around here, a translocation between chromosome 6 and 10 here. Now, it's interesting what we noted is that this translocation actually changed the pattern of the distribution of the repetitive sequence that I presented before, the satellite repeat 1 and the 169. In particular, this translocation based on the bioinformatic analysis on the VacCAP map, it should lead to have one chromosome that has both the chromosome, the satellite 169 and then the satellite 1. And then there should be one chromosome that do not have the satellite repeat, and then the other that have normal satellite repeats. So then we took this information and then we we verified the presence of these satellite signals using fish analysis. As expected, we found the chromosome that has the two signal, this chromosome, it's up here, that pair with a chromosome that has a normal satellite 1. And then we have a chromosome 6 that have a normal satellite 169 that pair with a chromosome that does not have a signal that it's likely this chromosome. So so this analysis verified the presence of this translocation. Recently, also what we did to add further verification of the translocation, we designed probe that are specific of the entire chromosome 6 and 10. And then what we verified is that there is one chromosome that has part of the chromosome 6 and part of the chromosome 10. And then there is another chromosome that is the same as a short part of chromosome 6 and a longer part of chromosome 6 and then a short part of chromosome 10. So it matches actually the expected pattern based on the physical map. So now, in terms of recombination, so this translocation actually should affect the recombination of the chromosomes. Indeed, what we observe is that some of the chromosomes have a long chain because they pair each other, because this translocation create a bridge between chromosome 6 and chromosome 10, and this is one example of a long chain of more than 4 chromosomes that make that chain during the meiosis. So then we used this is what we observe in the fish work. So then we used the linkage map to look at the presence of the translocation, and when we did the grouping analysis of the markers, what we were expecting is that there should be 8 groups, which are representing 4 homologous of chromosome 6 and 4 homologous of chromosome 10, that actually should not be able to separate in a linkage map construction. And that's what we observe. There are these 8 groups that don't separate, and there are 2 groups in particular that make the link between these 8 groups. And these chromosomes are actually these chromosomes 10-6 and 6-10. When we developed the linkage map, we could see that there is the translocated piece here. So we basically have multiple evidence that the translocation is there and does affect the pairing and recombination of the Draper genome. Next, we ask the question if other tetraploid cultivar have the translocation, and then what we saw is that none of the other cultivars that were included in the study have the translocation. This is the alignment between chromosome 6, W85 chromosome 6, and the Reveille by Arlen linkage map, and the Draper selection by Jewel linkage group. So we can see that here it's completely collinear, and they have a normal chromosome 6 and chromosome 6. Okay, and then also the fish work show that there is none of the chromosome that has the double signal with the satellite 1 and the satellite 169. So likely this translocation is not present in the other cultivar, but so far we found only in the Draper genome. So then we move the next to address another question about elucidating blueberry recombination behavior as being auto or allo tetraploid. Now, we know just a quick introduction about polyploidy and in particular related to tetraploid. So in general, we can have a polyploid species as like a tetraploid that can be autopolyploid or allopolyploid. So the definition of auto or allopolyploid depends on two factors, on the origin and the behavior of the polyploid. So which means that they have for each of the chromosome set, they have 4 copies, which in autopolyploid they derive from a genome that are highly similar, very low divergence. In allopolyploid, these genomes they come together are highly divergent. Now, like I said, the factor that play a role into the being autopolyploid or allopolyploid are two: the origin of the polyploid and the genetic behavior. So today, and in this work, we focused heavily on the genetic behavior, and we will look next to what we mean for genetic behavior. So first of all, in an autopolyploid, you have that the 4 chromosomes are highly similar, and they can the recombination or the inheritance of the DNA, it's random because they can independently pair and exchange DNA with all the other copy randomly. Where in an allopolyploid, these exchanges are preferential between the chromosomes that share more similarity. So then in autopolyploid, usually you can have recombination or pairing in multivalence because these chromosomes are highly similar versus in allopolyploid, this frequency of multivalent, it's very low and actually in natural allopolyploid, it's very rare. Now, as a consequence of the multivalent formation, we can have also double reduction, which is a phenomenon that is typical of the autopolyploids and lead to having a copy of the chromatid that are the same one, like here in this case, in the same gametes, and which can reduce the frequency of the heterozygotes. But this phenomenon is not present in allopolyploid. And there are ways with molecular markers today that we can trace these 3 behaviors that we have I've shown here. So why it's important to study if a species is an auto or an allopolyploid? Because this has a direct implication on trait inheritance, but also tell us which genetic model for genetic analysis we need to use. In case of autopolyploid, we need to use tetraploid models, and in case of allopolyploid, we use diploid model for genetic analysis. In blueberry, so there are a little bit of information before about the polyploid behavior. Very recent study based on cytogenetic working indicate that it's an allopolyploid. But most in early 2000 using molecular marker, there were indication that was an autopolyploid, although the number of markers used were not distributed genome-wide. And then recently, with the publication of the Draper genome, the analysis of the similarity between the homologous and transcriptome analysis suggested that blueberry has a allopolyploid origin derived from 2 closely related species. This does not exclude autopolyploid genetic behavior. So now, considering that tetraploid blueberry introgress multiple diploid species, it is possible that partial or allopolyploidy genetic behavior exists. So, but this question was not addressed at the genome-wide level, and that's what we addressed in this study. So the first thing we tested for preferential pairing, so random versus preferential, as highlighted in this scheme, and then we didn't find any evidence of preferential pairing for any of the chromosome, so which suggests that all the chromosome recombine or in a random as a polysomic organism. Next, we tested the pairing and multivalent formation using molecular marker and also the fish work. Here it's the scheme of the multivalent formation, and then we noticed that there is a significant amount of quadrivalent formation in all the linkage map, all the sample that we evaluated were included in the linkage maps. Was a little bit lower in the derivative biloxy, but these were also the linkage map that has had the lowest number of markers. The cytogenetic analysis also highlighted the presence of these rings, which are the multivalent chromosome pairing. So definitely there is a relatively high significant frequency of multivalent formation that is typical of an autopolyploid. Then next we evaluated the presence of double reduction, and then we detected a significant fraction of double reduction across all the chromosomes. So overall, the conclusion of this work highlight a new high-quality reference genome named W85 that has been released and complement the already available driver genome. The discovery of this and the translocation, verification of the translocation in the driver genome that affect chromosome recombination and pairing and can impact trait inheritance. And we demonstrated that blueberry genome behave as an autopolyploid, and which I want to highlight that this does not exclude a possible allopolyploid origin between from two closely related species. This is work that will be looked into in the future. With that, I want to just highlight that as part of the project, we have been, so the blueberry, the reference genome, the W85 genome has been used as a reference for the pan-genome and the genotyping platform that we have been developing as part of the VacCAP project. Another 6 genomes have been released in the last couple of years. And so far we are seeing that there is a high level of collinearity between the genome. Now, overall, these genomic resources are enabling us to advance gene discovery, and this is just some example of genes in the anthocyanin pathway that we have been identifying in a blueberry or cranberry, thanks to this larger number of genomic resources that we have generated. So going back to the W85 genome, the genome is released on the GDV, which is maintained by Dr. Bain Lab and managed with help from Jody. You can see here the W85, the 20 haplotype 1 and haplotype 2. Then you can go to the link, and then here are all the future analyses that are available for this genome. So then I want to acknowledge all the people that have worked on the project. Dr. Mengist and Boston have been working largely on the genome assembly and characterization. Dr. Boston and Mengist on all the genetic analysis. The cytogenetic work was done by the group of Marina Iovine from Italy and his collaborator. Repetitive sequence analysis was performed by Dr. Patrick Edigar at Michigan State University with his team. Then acknowledge all the collaborators that have provided material and data for the linkage maps. Also, inputs from Nikolai Vorsa. Dr. Vorsa was the first one that had developed and selected the W85 clones accessions, and Jim Bangkok from some input on some of the germplasm that was used. Of course, knowledge to the NIFA for the funding on the VacCAP. Special thanks to Dr. Chad Finn, who started this, was part of the project and this work at the beginning with providing some of the material used for the linkage maps. And I want to thank you for your attention, and look out for the January 5 issue on New Phytologist, where we will have the article featuring the cover of the new phytology issue. With that, I will take any question.
Massimo, thank you so much for a great webinar. Fantastic information that you share with everybody, and we are really looking forward to that new Phytologist publication. I just want to thank everybody for joining us. Before we take questions, I just want to remind everybody that there will be a follow-up survey, your favorite. Please help us with your feedback to improve our webinars. And so if you have a chance and some time to answer that survey, we would really, really appreciate it. Again, as I said before, and it's in the chat, but I'll put it again, the recording of this webinar will be in our YouTube channel for the VacCAP project. And it's also going to be, there's going to be a link to it in our website. So I'll put both of those links again on the chat. And with that, we also want to advertise our next webinar. We will have another webinar coming up on January 26th. We are very fortunate to have Dr. Nala Basil, who will be talking on this webinar on the 16th on 2 new flex sequencing EXL high throughput genotyping platform for blueberry and cranberry. So if you're interested, just there will be a link to register in our website, but also we will be sending out information. If you get our newsletters and if you get our post on Twitter, you will also see a link there. So look out for that email. So with that, there's a couple of questions in the chat. Massimo, I don't know if you were able to look at them, or if you want to go ahead and answer them.
Yeah, I looked at them. So partially Michael has already addressed one of the questions, but yes, and I confirm what Michael said, that it's really compatible with the tetraploid chromosome if through true gametes, unreduced gametes. So have you compared the collinearity between this genome and vaccinum Darwai? Can this genome be used as a reference for mapping vaccinum Darwai sequence reads? So to address the first question, yes, there is a slide that I didn't show, but yes, it's highly collinear with the completely collinear with the Darwai genome. Can the genome be used as a reference for mapping Darwai reads? We didn't do mapping the reads. I'm sure that you will find some level of similarity or dissimilarity, but we aligned the genome, and the genome can be are highly collinear. But again, no, we didn't use it for mapping reads from Darwai. Which Darwai accessions was used? I'm assuming the question is about the Darwai accessions for the fish work. We used the one that was available at the repository, and was the one at the repository, which confirmed the results of the alignment with the genome, that the Darwai genome is not a source of the translocation. So we also looked at Darwai as a possible source of the translocation, and it's not.
I think Nala has her hand up. I think she wants to make a comment.
No, I have a question. Great job, Massimo and team. Thank you so much for this informative webinar. My question is, did you look at whether the translocation is present in the parent of Draper, Duke, or in any of the offsprings?
We are just in the process. It's not in Duke so far. We don't have strong evidence that it's in Duke.
Do you have the other parent, Massimo?
I do, but it's not available.
It's not available. We don't have it.
I already checked. With you, and we see that it's not available. Yeah, so we are doing some work to see if we can find a way to trace that translocation. But not having the parents, it's a little bit challenging.
So, but the offsprings, are you looking into that, in the Draper by June population?
Yes.
Okay.
We do have some of that.
Okay.
But in the Draper by Jewel, it's there because we know it's there because it's in the linkage map.
Oh, that's right. Okay.
Yeah.
So, okay.
Yeah.
Thanks, Massimo.
Welcome.
Okay, it looks like we have another question. Go ahead.
Yes. Hi, Massimo. This is a great presentation. I have one quick question with regard to, since we have the fish analysis and all, is there any percentage of that we can expect of reduced gametes from a cross between diploid derivative with tetraploid chromosome?
If we should expect that there are cross unreduced gametes, so unreduced gametes in diploid blueberry wild species, especially Darwai and Caesariensis, it has been observed in a relatively high frequency if you compare with some other species, and that's why it's easy in blueberry to generate interspecific crosses. In other systems, like I worked in potato in the past, it's not easy to have many diploid species that have two gametes, and then you need to do a lot of lab work to double the diploidy of those wild species to bring them to the tetraploid level. But in blueberry, it's relatively easy. So, yeah.
So I don't want to pick on it, but is there any expectation that we can predict from the fish analysis to the how much the expected I don't know this kind of study is ever published or not, just out of curiosity?
No, I mean, relative to the fish analysis we really didn't look at that question, but there is literature where people have looked at cytogenetic analysis of wild species, diploid blueberry wild species, where they looked at two end gametes formation, and yes, it's relatively at a high frequency, so it's possible.
We have two other questions on the chat. So Jim is asking, do you see any pollen abortion due to translocation?
So we started to address the question. Part of the results are in the publication. We didn't see any pollen. The pollen viability seems to be normal so far.
Okay, and then Hamid is asking, what is the significance of this transformation? Are there any genes related to any particular traits on it?
No, on the paper, we did not look at, it's not like we didn't do any genetic analysis for any traits. The significance is the translocation, yes. So that's another topic that we are trying to follow. Technically, there is a major QTL in chromosome 10 in blueberry that could be affected. It's regulating Fritz tolerance, that it's in chromosome 10. Now, trace that effect of the translocation, it's another story. It's yes, it's a project itself, and but will be interesting to follow how these affect trait inheritance.
We have one other question. This is, what are the challenges for gene fusion novel transcript discovery in blueberry? How those can be overcome while analyzing blueberry RNA sequence data?
Okay, so I guess, Michael, isoseq data, but we didn't look into that in this analysis. But probably the best will be to do some work with isoseq and then verify those gene fusion. I know that there is some work ongoing from the blueberry community, but I would suggest say isoseq data from tetraploids to look at those events.
Great, Massimo, thank you so much for answering all the questions and everything you share with us today. We're getting to the end. Oh, we have one more question. We're just going to be very generous today, and we're going to let Michael go ahead. Michael, if you want to just unmute yourself.
Yeah, I'm just curious, when you're looking at the potential for an allopolyploid origin are you going to be doing that analysis with wild tetraploid chromosome, or are you going to be doing that analysis looking at the cultivars? Because I would think that if you're just looking at cultivars, that since all of the cultivars represent introgressions from different species, that would be really hard to decipher.
Yeah, Michael, I have to admit that I am much more interested on the behavior, because, you know, tracing the ancestry might be a little bit, I mean, that's the thing, that blueberry might be it's probably an interesting model, especially for studying the boundary between allopolyploid and auto, and like, you know, genome dominance, effect of interaction between genes, because it's possible that are two very, very closely related species that crossed, but they behave like an autopolyploid. Okay, so I think that, yes, more comparative analysis with diploid species will help. More genome dominance study using transcriptome data and trace some of this effect. But like I said, again, it does behave like an auto in terms of genetic inheritance, and yeah, so more comparison with wild species definitely will be helpful in the future with more cultivars. Okay.
Thank you.
And I want to just make sure that I highlighted all those cultivars that we used in the linkage map construction. They all introgress wild species. So if there was some signature of preferential pairing in some of them, we would have detected. In terms of genetic behavior, they do recombine like an autopolyploid. So
great. So we're getting to the end of our webinar. Again, Massimo, thank you so much for sharing all this information, and to all the participants for their questions. Again, this is going to be posted on our YouTube channel if you want to go back, or if you want to share it, and we're going to share it on our social media as well. And thank you, and look forward to our next email for our webinar with Dr. Basil in January.
Thank you so much.
Bye-bye, everybody.
Bye-bye.